السلام عليكم ورحمة الله وبركاته. حياكم الله جميعا. أنا محمد الحمود، طالب في السنة الثالثة بإذن الله، وحاب أقدم لكم Introduction to Neurophysiology. قبل ما نبدأ حاب أقول لكم أي شخص عنده سؤال أو في نقطة ما كانت واضحة حاب أعيدها وأوضحها أكثر، either raise your hand أعطيك permission to speak، أو write it in the chat box أو in the Q&A box. أنا شايفهم كلهم خلال المحاضرة. بسم الله. فؤاد طلب مننا نراجع الكرينيال نيرف في كل محاضرة، بحيث أنكم يعني لو ما طلعتوا من الكورس هذا إلا وأنتم حافظينهم أو حافظين معظمهم. So I'll say the number and the name and briefly the function of each nerve. So the first nerve is the olfactory nerve. It's involved in smell. The second nerve, the optic nerve, is involved in sight, vision. The third nerve and the fourth nerve and the sixth nerve are all involved in eye movement. The fourth nerve, which is the trochlear nerve, is involved, the fifth nerve, which is the trigeminal nerve, is involved in mastication, which is chewing, facial sensations, and the general sensation of the tongue. You know that in your tongue, you can taste and you can feel things like pressure and touch. So when we speak about touch or pressure, that's somatosensation or general sensation. And then there is taste, which is felt by another cranial nerve, which is the facial nerve. So let's go through the first six nerves again briefly. The olfactory nerve, smell. The optic nerve, second, sight. The oculomotor nerve, third, eye movement. The trochlear nerve, fourth, eye movement as well. The trigeminal nerve, fifth, chewing, facial sensation, and general sensation from the anterior two-thirds of the tongue. The sixth, the abducens nerve, eye movement. Then the facial nerve, which is the seventh, is involved in facial movement and taste of the anterior two-thirds of the tongue, lacrimation, salivation, and many other functions. The vestibulocochlear, which is the eighth nerve, is involved in hearing and balance. The glossopharyngeal, which is the ninth, is involved in taste, sensation, and general sensation from the posterior one-third of the tongue. So, as we've just mentioned, that facial is involved in taste sensation from the anterior two-thirds, and trigeminal is involved in the general sensation from the anterior two-thirds. The glossopharyngeal does both functions to the posterior one-third of the tongue. It's also involved in monitoring the carotid body and sinuses. The vagus nerve, as Fuad discussed, is involved in many organs. It's the 10th nerve. The accessory nerve, which is the 11th, is involved in head turning and shoulder shrugging. And finally, the hypoglossal nerve, which is the 12th nerve, is involved in the tongue movement. That's not the topic of our lecture, but we wanted to review it quickly for you to memorize it. So what is our lecture outline? We're going to talk about the divisions of the nervous system, the spinal cord reflexes and the reflex arc, synapses and synaptic transmission, and neurotransmitters. So to understand neurophysiology, it's important for us to appreciate the organization of the nervous system in both terms of anatomical division and functional division. So anatomically, the central nervous system is divided into the central nervous system and the peripheral nervous system. The central nervous system, as you can see highlighted here, involves the brain and the spinal cord. It's the most complex part of the nervous system, as it contains the majority of nerve cell bodies and synaptic connections. The peripheral nervous system, on the other hand, includes nerve endings or receptors, nerves, and ganglia. And it basically constitutes a link between the central nervous system and the peripheral organs. So in both terms of sensation and motor functions, it takes the sensory information from these organs, delivers it to the central nervous system, and takes any commands, any motor commands from the central nervous system and delivers it to the organs. So let us take a look at the anatomical division of the brain itself. So the brain consists of many parts, as I discussed. It includes the spinal cord, the brainstem, which is made up of the midbrain, the pons, and the medulla, the diencephalon, which is made up of the thalamus and the hypothalamus, the cerebellum, and the cerebrum. So let us take a deeper look into each of these structures. So starting by the spinal cord, you can see it here from a lateral view and a cross-section of it right here. So it's basically a long tubular structures, as you can see. It's like a tube. And it's protected by the bony structures of the vertebral column. It includes a central canal that is not shown here, but I believe you've seen it in Fahad's presentation, and is protected by the meninges, like the brain. So in these meninges and inside the central canal runs the cerebrospinal fluid, which also runs inside and around the brain to provide protection to them. So information in the spinal cord travels up and down in tracks. As you can see here, in the center, there is this grayer portion, which is called the gray matter. And on the outside of it lies a white portion, which is the white matter. And as Fuad discussed with you, gray matter is made up of cell bodies, while white matter is made up of axons or fibers. So in these gray portions lies the anterior horn, the lateral horn, and the dorsal horn, which all contain cell bodies that do various functions. And then in these white portions lie the tracts, which are divided into ascending tracts, which are basically sensory tracts that deliver information from around the body into the central nervous system, and descending tracts, which take motor commands from the central nervous system and delivers them to various parts of the body. Now we take a look at the brainstem. So the medulla, the pons, and the midbrain together are called the brainstem. 10 of the 12 cranial nerves that I just revised with you arise from the brainstem. Only the first two, the olfactory and the optic, arise from different places. The rest all arise from here. So what are these structures? The medulla, which is the most inferior part, is involved in blood pressure regulation, breathing, swallowing, coughing, and vomiting. You took a look at it in respiratory block, as I remember. The pons is involved in facial expression, sensation, and equilibrium, as it connects the cerebellum with the cerebrum to facilitate their functions. The midbrain is involved in vision and hearing, and it contains the centers for both vision and hearing. Let us take a look at the difference between the cerebrum and the cerebellum. So the cerebrum, al-mukh, cerebellum, al-mukhaykh, are different, as Fahad illustrated. They both involve like leafy structures where there is rises and like these gyri and sulci. However, they're here called a different name. They're called folia here. So that's just something that you will learn along the way, but I just felt like sharing it. So it's responsible for coordinating, planning, executing movements, and as Fahad mentioned, it's the maestro of the central nervous system. It coordinates the different parts of the nervous system in order for them to perform their functions well, and in terms of proprioception and other functions as well. The diencephalon, which lies above the brainstem, is made up of the thalamus and the hypothalamus. The thalamus is basically a relay station. Okay, what's a relay? A relay is basically a synapse, which is when a neuron comes in contact with another neuron to deliver neurotransmitters to it, either to excite it or inhibit it. So the thalamus is where most most ascending tracks end up, and most descending tracks come by while descending to the spinal cord. So most of the information that goes to the cerebral cortex or the cerebrum pass by it, and most information going down from it pass by it. The hypothalamus, which lies anterior to it, is involved in homeostasis, and it's linked to the endocrine system, especially to the pituitary gland, as it controls its secretion of hormones. The cerebrum includes both the cerebral cortex, the deep nuclei, which are the basal ganglia, the hippocampus, and it involves also other structures such as the ventricular system. It is basically the part that is involved in perception and higher motor functions, and basically the part where we use to perceive information. So it's divided into two lobes, the left and the right lobe, as you can see here. They're connected by a band of fibers called the corpus callosum. And its cortex, the cerebral cortex, includes four lobes: the frontal lobe, the parietal lobe, the temporal lobe, and the occipital lobe. These lobes have different functions and are divided by fissures or sulci: the central sulcus here, the lateral sulcus here, and the parieto-occipital sulcus here. Each of these lobes is subdivided into primary areas, secondary areas, and association areas. And each of these areas is designated a number that correlates with its level of involvement in the task that it does. So the primary motor area, for example, which lies here, anterior to the central sulcus, is involved in the voluntary movement. And other areas such as Broca's and Wernicke's areas, for example, in the parietal lobe, are not primary areas, but secondary areas that are used to, in case of Wernicke's, to perceive what is heard, and in case of Broca's, help in forming words. So these are secondary areas, Broca's and Wernicke's, that involve more synaptic connections and are less directly involved with the task at hand. I'm sorry if I confused you, but hopefully you'll get a deeper look at all of these structures in the coming lectures with my colleagues, inshallah. This is just an introduction. The ventricular system of the brain. So as Fa'ad discussed with you, there are ventricles, and there are the meninges, between which run the cerebrospinal fluid, which helps in nourishing and protecting the brain and the spinal cord. So we have two ventricles within the parietal and frontal lobes. They're called the lateral ventricles. Between them run an interventricular foramen, which connects them, as you can see here. Then there is the third ventricle between the cerebral cortex and the brainstem. Then there is the cerebral aqueduct, which runs through the midbrain. The fourth ventricle, which is between the cerebellum and the brainstem, and then the central canal of the spinal cord, which we discussed a while ago. So that was the brain. All of these parts are all parts of the brain. Now, let us take a look at the peripheral nervous system. So the nerve endings, the nerves, and the ganglia. As you can see here, there are multiple nerves exiting both the brain and the spinal cord, forming plexuses here, for example, the lumbar plexus and the brachial plexus, and extending to the tips of fingers and the toes. So what are these peripheral nerves? So it's made up of nerve endings, which include sensory receptors that detect changes in the internal and external environments in our body. It includes afferent ending, so a receptor that perceives a sort of input, then an afferent neuron or a sensory neuron that delivers this type of input to the place where it should go. For example, the sensation of the hands should go to the specific place in the parietal lobe which is involved in the sensation of the fingers, for example. The peripheral nerves both include the cranial nerves, which are the 12 cranial nerves that we revised, and the 31 pairs of spinal nerves that exit from the sides of the spinal cord here. These are the afferent fibers and the efferent fibers. Then there are the ganglia, which are basically structures that are outside the central nervous system, where nerve cell bodies exist. An example for them would be the dorsal root ganglion, which is involved in the afferent fibers of the spinal cord. So we took a look at the anatomical division of the nervous system. Let us take a look at the functional division of it. So functionally, it's not divided into central and peripheral. It's divided into sensory or afferent and motor or efferent. The sensory branch is involved in delivering or bringing information from all around the body into the central nervous system. And it includes many receptors such as visual receptors, auditory receptors, and somatosensory receptors like those involved in touch, and many other types of receptors that the information which these receptors take go to the central nervous system. Then there is the motor branch, or the efferent branch of the nervous system, that is involved in delivering a response to those receptors, to what the sensory division brought. So it involves carrying information out of the nervous system into the periphery or peripheral organs. So, for example, it's involved in contraction of muscles, secretion by glands, and many other functions. This motor or efferent system is divided into a autonomic. This motor division is divided, it is subdivided into a somatic nervous system and an autonomic nervous system. The somatic nervous system is under our control. We voluntarily control it. It's usually involved in the control of skeletal muscles, like raising our hands. And it usually only involves a single motor neuron. So it has a basic circuit. So this is the neuron cell body in the central nervous system. This is its axon, and it directly affects the organ that we're trying to move. Then there's the autonomic nervous system, which involves multiple synapses until the neuron reaches the organ that we're speaking of. This autonomic nervous system, which is involuntary, unlike the somatic nervous system, so we cannot control our autonomic nervous system. Its control is dependent on its receptors throughout the body. So this autonomic nervous system is divided into the sympathetic and the parasympathetic nervous systems, which you've been exposed to before. So I'll just mention a few points here that are a bit important, but you know most of the information regarding the autonomic nervous system as you've already studied it. So the sympathetic nervous system, which is usually referred to as the fight or flight response, is involved in preparing the person who is in a stressful situation to be able to escape it. So the heart rate will be increased, the arterial pressure will be increased in order to increase blood flow to the muscles. The blood sugar will increase in order to supply the muscles, and the mental activity or alertness will also increase. While on the other hand, the parasympathetic nervous system, which is referred to as the rest and digest response, is involved in energy conserving and processes like digestion and eating. So what are the important points to point out or differences to point out? The sympathetic division, it originates in the thoracolumbar segment of the spinal cord. So from T1 to L3, while the parasympathetic originates in both the cranium, the brain, and the sacrum. So the four nerves, the third, the seventh, the ninth, and the 10th are all involved in parasympathetic, in the parasympathetic division, and S2, S3, and S4 from the sacral segments are also involved in it. These are differences that you can read later, like the length and the location of the autonomic ganglia. So let us take a look at spinal cord reflexes. Spinal cord reflexes are basically, let us actually, before speaking about the spinal cord reflex, let's take a look at the reflex arc. Now, if you pinch your skin, the receptors on the skin are going to sense it. Then the information will be carried through a sensory nerve to the integration center, which could be in the spinal cord or in the brain. Then the brain will decide that since that thing pinched you, it would be wise to remove your hand from it. So it'll command the motor neuron to go and contract a specific muscle or relax a specific muscle in order to push it away from this harmful situation. So these are the components of the reflex arc or the reflex structure. So it starts with a receptor, a sensory neuron, a modulatory integration center, a motor neuron, and an effector organ. So There are three common types of spinal cord reflexes. Spinal cord reflexes are basically reflexes that are not facilitated by the brain. They are completely integrated within the spinal cord. They include the stretch reflex, the Golgi tendon reflex, and the flexo-withdrawal reflex. Each of them has a specific complexity and different stimuli and different responses. So an example for the stretch reflex is the knee jerk reflex, which is basically if you kick a, have you seen a doctor? Actually, let's take a look at it here. Knee jerk reflex. I should have added a picture, to be honest. Yeah. So basically, a knee jerk reflex is when you hit with a hammer the patella tendon so that it stretches the quadriceps. As a result, it will contract and you'll see the quadriceps or you'll see a kick happen. So that's a reflex that involves only one synapse. It was stimulated by the stretch of the muscle, the quadriceps muscle, and the response was the contraction of this muscle. There is the Golgi tendon reflex, which is when a muscle is over contracted. The response would be the relaxation of that muscle. And there is another reflex called the flexo-withdrawal reflex, which is an example of which is when you touch a hot stove, you'll feel the burning sensation. So the pain or the temperature will be the stimulus. And the response would be flexing that side, pulling back your hand, and extending the other side. It involves many synapses, and it is the most complex of these three types. Let us take a look at synapses and synaptic transmissions, as we've spoken about synapses and synaptic transmissions more than once throughout this lecture and throughout the lecture with Fuad. So there are two major types of synapses in the body. There is a chemical synapse and an electrical synapse. The chemical synapse, which is most of the synapses in the central nervous system, it's basically a synapse where the first neuron secretes a certain neurotransmitter that acts on receptors in the second neuron. An electrical synapse is where the two neurons are connected, or the two cells. The first could be a neuron and the second could be something else. They're connected with proteins, gap junctions that allow the free movement of ions between these two cells. So I won't go into details in the electrical synapse, but I'll go into details in the chemical synapse since it's the most common. So we can see here this presynaptic neuron, or called the presynaptic terminal, or the presynaptic knob. And here is the postsynaptic cell, or the postsynaptic terminal neuron. Between them, there is this synaptic cleft, which is a small space between the two neurons. If we take a look at the presynaptic neuron, we can see that there are mitochondria, which produce ATP that is involved in the production of these neurotransmitters and in putting them into these vesicles. And then there's these synaptic vesicles, which include the certain neurotransmitter that is going to be produced here. Then we can see calcium channels, ion-gated calcium channels that I'll talk about in a bit. So when these neurotransmitters exit the cells, when an action potential arrives at this synaptic terminal or presynaptic terminal, synaptic vesicles will be exocytosed into the synaptic cleft. These synaptic vesicles include the neurotransmitters, which will work on these receptors on the postsynaptic cell. They'll either excite it, inhibit it, or modulate it. So in order for that calcium channel to open, it's about the voltage, the membrane potential of the cell. So when an action potential arrives and reaches the potential required to open the calcium channel, this voltage gated calcium channel will open and allow the influx of calcium. Calcium is a positive ion, so it will depolarize the cell. And as the result of depolarization, the synaptic vesicles will be released. So we talked about different types of, we talked about the process of exocytosing these vesicles into the cleft. How about the process of acting on receptors? Okay, the synaptic vesicles were exocytosed. This neurotransmitter is now in the synaptic cleft. How does it work on the next cell? So on the postsynaptic cell, there are two types of receptors. There are ionotropic receptors or ion channels, which are channels that allow the flow of ions, either cations or anions, and are opened by a ligand or basically a neurotransmitter. And there are metabotropic receptors, which are also activated by a signaling molecule or ligand, but work in a more complex way. Let us take a look at it in here. I hope you can hear it. Tell me if you don't hear it.
2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss receptors and ligands. When neurotransmitter molecules are released from a neuron, they pass the signal to the next neuron by interacting with receptors on that neuron. These receptors are made up of proteins embedded in the membrane of the postsynaptic cell. There are two broad families of neurotransmitter receptors. One type are called ionotropic receptors. Ionotropic receptors have a site where a neurotransmitter can bind called the binding site. When a neurotransmitter binds here, it causes a channel to open in the receptor, and ions are permitted to flow into the neuron. This can change the membrane potential of the neuron and send a signal known as the action potential down that neuron. The neurotransmitter that binds is called the ligand, which is just a term for any substance that can bind to a target protein. Ionotropic receptors are also called ligand-gated ion channels. The other type of receptors are metabotropic receptors, also known as G-protein-coupled receptors. Neurotransmitters also bind to these receptors, but instead of immediately opening an ion channel, the next step after binding is the activation of an intermediate protein called a G-protein. The G-protein can then influence the opening of ion channels, but it can also affect enzymes and activate intracellular signaling molecules known as second messengers, which can initiate signaling cascades within the cell. G-protein-coupled receptors thus tend to have a slower action, but can have more widespread effects due to their ability to influence various molecules throughout the cell. Ligands other than neurotransmitters, such as drugs, can also bind to receptors and have a variety of effects. If they have the same effect as the neurotransmitter, they are known as agonists. If they block the effects of the neurotransmitter, they are known as antagonists. Some drugs can bind to the site the neurotransmitter binds to and have the exact opposite effect of the neurotransmitter. These are called inverse agonists. Finally, drugs may bind to a site on the receptor that is separate from the site where the neurotransmitter binds and affect the likelihood that the neurotransmitter will bind. In this case, they're called neuromodulators and said to have an allosteric effect.
Okay, I hope that explained the difference between the two types of receptors. So,
welcome to 2-Minute Neuroscience, where
So moving on to the postsynaptic potential. Okay, so a certain neurotransmitter worked on the postsynaptic cell and activated a certain receptor. The effect can either be excitatory or inhibitory. Excitatory when the cell is depolarized, and inhibitory when that cell is hyperpolarized. So for excitatory postsynaptic potential, there are neurotransmitters, excitatory neurotransmitters that cause it, such as acetylcholine, norepinephrine, and epinephrine. Sorry. Excitatory neurotransmitters like acetylcholine, norepinephrine, and epinephrine, glutamate, serotonin, and dopamine, all of these neurotransmitters cause an excitation of the postsynaptic neuron. While other neurotransmitters such as glycine and GABA or gamma aminobutyric acid cause an inhibition of this postsynaptic membrane or postsynaptic neuron. So let's take a look at neurotransmitters. There are excitatory and inhibitory, as I've just given you an example. And there are those who do not excite nor inhibit themselves, but modulate the function of one that does. So sometimes a neurotransmitter can enhance the function of another or inhibit the function of another without acting on its own. So here is a list of certain neurotransmitters with their functions. For example, acetylcholine is involved in motor functions, attention, arousal. It's for you to read whenever you have the time to. Now to the most exciting part, I guess, of the lecture. Let us do a case study based on something that we have studied in this lecture and something that you guys have been exposed to in the previous year in the MSK block. Is there anyone who would like to read this case? You can simply raise your hand if you want to read it. All right, so an 18-year-old college woman comes to the student health service complaining of progressive weakness. She reports that occasionally her eyelids droop and that she tires easily, even when completing ordinary tasks such as brushing her hair. She has fallen several times while climbing a flight of stairs. These symptoms improve with rest. The physician orders blood studies, which reveal elevated levels of antibodies to acetylcholine receptors. Nerve stimulation studies show decreased responsiveness of skeletal muscle on repeated stimulation of the motor neurons. Does anyone know the diagnosis here? Exactly, Ahmed and Hannah, myasthenia gravis. So what is the explanation? This young woman has classic myasthenia gravis, which in the autoimmune form in the disease, antibodies are produced to acetylcholine receptors on the motor end plates of skeletal muscles. So her symptoms of severe muscle weakness in the eye muscles, arms, and legs are explained by the presence of antibodies that block acetylcholine receptors. Although acetylcholine is released in normal amounts, the binding of acetylcholine to its receptors on the motor end plates is impaired. Because acetylcholine cannot bind, depolarization of the motor end plate will not occur and normal action potentials cannot be generated in the skeletal muscles. So can anyone tell me how will we treat this patient? Ella, do you guys remember? We took it in MSK. Yes, acetylcholinesterase inhibitor. Perfect. So the treatment is basically dependent on understanding the physiology of the neuromuscular junction. So This patient's condition improved with administration of pyridostigmine, which is a long-acting acetylcholinesterase inhibitor. The success of the treatment confirmed the diagnosis of myasthenia gravis. Acetylcholinesterase inhibitor on the motor end plate normally degrades acetylcholine. So inhibiting this degradative enzyme will allow the acetylcholine to its levels to stay high and work on the few receptors that are not affected by the acetylcholine receptor antibodies. So let us take a brief look at it. So acetylcholine is produced normally here. The receptors here are blocked by an antibody. An enzyme here called acetylcholinesterase degrades acetylcholine after its release so that it does not stay in the synaptic cleft and occupy it. When we inhibited this enzyme that degrades it, the few receptors that were not attacked by this antibody are now functioning. The function will not be regained completely, as there will still be some receptors that are blocked by these antibodies, but at least more acetylcholine will reach the receptors that are not affected. So that was it for the lecture. Here are the references that I used. And if anyone has any questions, please feel free to raise your hand, write it in the Q&A box or in the chat box. Thank you all for attending. I'm really sorry if I confused any of you with any of the complicated parts that we've mentioned, but my colleagues are going to present you guys with lectures on pretty much each of the structures that I've mentioned. So don't worry, and inshallah you'll grasp it all in this week. If not, you have a whole block to understand it in. There will be a lecture in about 15 minutes. I think it's about CNS infections. Let me check. Yeah, in 15 minutes, Nir Al-Hadlik will introduce CNS infections to you guys. So good luck with that lecture, and good luck with the block.